I come from Zhejiang Agriculture and Forestry University. Today my topic is about identification by deep sequence and profile of conserved and novel hickory microRNA involved in graft process. There's several parts I should give presentation. First, I have to introduce hickory in briefly. In fact, hickory is a kind of walnut cheese. It is very well known in nut cheese in eastern China, and mainly found in the area of Tian Mountain. This is a nut, hickory nut, and this is the hickory tree. And we collected 155 genotypes from Zhejiang, Anhui, Hunan, and Guizhou and Yunnan province. So there are several provinces we collected. In fact, in China, there are 5 Chlea species which is native to China. So this is a species called Chlea casayensis, and Dabiasanis, and Hunanis, and Turkinis, and Kurchunis. So hickory, in fact, is very important economically in China because there are a lot of farmers, they grow this kind of walnut, and because this nut is very good sources for oil and nutrition. So so in the table 1 we can compare the oil content among different oil cheese. So you can see here, this is hickory cheese, the nut of hickory cheese. The oil content is about 64 and 74%, and the unsaturated fatty acid is more than is about 90%. But this species, there are many main limitations is as following. The first one is lack of a new cultivar, and the second one is difficulty in propagation. The third one is it is slow growth and later maturation. The fourth is there's low yield and poor quality. So at first we have to do some research with morphological and physiological characteristics. So we can see here we use grafting technology to get good quality and hickory nuts. So we can see, and when we use root stock and sign and to measure the fetal hormones. So we found that the auxin plays a key role in the grafting process in hickory, and we also use immunocolloid technology to detect the auxin. So we find that it is located in the plasmid. And here shows the grafting technologies to grow hickory. So it shows that we use normal technology, and the second one we use auxin applied plant, and C is we use auxin inhibitor NPA. So we compare this several technology to find which genes is related with them. So and in physiological levels, we find that the types of sign and graft times and is main factors that affect survival rate during grafting process. I'm sorry, so there's some Chinese. we know microRNA is very important in biological levels. So we try to test microRNA in hickory during grafting process. So in table 1 shows that this is statistical analysis of small RNA from grafting 0 days and 7 days, 14 days libraries. So we can see this is the 0 days library. We find there's several gene microRNAs we have get it, and this is 7 days and 14 days. And we statistic the microRNA library from 3 different times library in hickory. So we find that 24 BP base pairs, small RNA readers, there's largest numbers, they are largest. And we also get analysis and find that the conserved microRNA identified in hickory, there are 21 conserved microRNA belonging to 13 microRNA families, and of which 9 microRNA stars was detected, and which had strong evidence of being benefit microRNA. And we also get 10 novel microRNA, and their corresponding microRNA stars belong to 8 families during grafting process in hickories. And in table 4, we test the different replacing microRNA during graft process in hickories. So lots of them are more than 2 times. And we also use real-time RT-PCR to conserve the microRNA sequence. So we use 14 microRNAs, and leg placing chains were similar to the results obtained by selected sequence. And the targeted genes of microRNA, and we also tried to test, and we found the 4 microRNA targeted genes to perform real-time RT-PCR, and the results also shows that an inverse relationship between expression of microRNA and the regulation of their corresponding targeted gene. So this evidence further validated the reliability of microRNA sequencing generated by selected sequencing. So I have to get a conclusion. The first one showed that we got 155 genotypes from different provinces in China, and there are 21 conserved microRNA sequences, as well as 10 novel and 10 potential novel microRNAs were identified during the grafting process in hickory. And that's actually thanks the Nature and Science Foundation of China and Zhejiang Province Nature Science and Foundation, and also we have to thank my students. Thank you all.
Okay. Do we have any questions, folks?
Hi. My question is, previously we just think the microRNA is a kind of non-coding RNA, but recently more and more papers just published recently showed, in fact, the pre-microRNA they have some ORF or open reading fragment. That means it can have some it can express the protein. And I remember Nature article just published several months ago showed its effect on the growth on some plant. In fact, I'm not the question. I'm just a simple comment. Do you have any idea about this?
In fact, I haven't read that papers. Maybe you can tell me that papers, and then maybe I can read it. Thank you. Thank you very much.
Thank you.
Okay.
Thanks for presentation, Dr.
Okay,
Zheng. So in the first part of your talk, you talk about the auxin response for the, and the second part, you talk about the graft process.
Yeah.
And how do you think the microRNA species you characterized corresponding to auxin response or not? Such as, perceiving auxin ligands or signal transduction?
Yes. In fact, tomorrow I will give a presentation which is related with auxin transport and auxin synthesis related associated genes, and I will test. There are some microRNA targeted genes is related with auxin associated genes. So I will test maybe in the future, near future. Yes.
Great.
Yeah.
Thank you.
Thank you.
More questions?
You looked at auxins in the grafting process, but did you look at any of the other plant classes of hormones, the cytokines, gibberellins, et cetera? Because auxin is, if I remember correctly, it's produced by the growing tip and prevents branching, and maybe that has to do with the significant growth, where other things like cytokines are involved in branching and may also be involved in the graft process.
I'm sorry, I can't catch what you said. You mean the auxin is more important than the other?
There are 5 groups of plant hormones. I don't plan to list all
Yeah,
of them.
there are 5.
Auxins are just one.
Yeah.
Auxins are produced by the growing tip of plants. Cytokines are produced by the roots. Okay?
Yeah.
And so I wonder if the lower part of your graft is also influencing the graft.
Yes. Yes. In fact, auxin can transport it from top of the leaf and top of the bud and go down into the base part, and when we do grafting, we have to cut the sign and cut the shoot, yes, and put them into the root stock. So auxin transport was inhibited. Is inhibited. How to say? So free auxin cannot be transported from the top to the base. So the banded auxin will be so the band how to say so in the auxin in the plasmid will be get free, and then the content of auxin in the rootstock and the sign will be increased. So auxin can can promote cell proliferation, and then let the grafting process continue. Yeah, thank you.
Thank you for your talk.